Today we're going to talk about a virus family that causes a very, very devastating disease, and we're going to talk about the virus that family that includes the virus that causes rabies. This virus is in a large group of viruses that actually is an order of viruses. So this is an example of a virus family that is found within an order. And this order is called the Mononegavirales. Viralis is the indication for order of viruses. And all of these viruses, even though these are RNA viruses and have a pretty high rate of mutation, there's enough similarity in the way that these viruses within this order replicate that has placed them together. So we're going to be looking at a group of related negative strand RNA viruses. The order gets its name, mono from single, nega for negative, and viralis for the viruses, virus order. Within this order, there are 4 virus families. The first one is Rhabdoviridae, second Filoviridae in no particular order, the third is Paramyxoviridae, and the fourth is the subject of today's lecture, which is Rhabdoviridae. We will talk about Filoviridae and Paramyxoviridae later on at the end of the class when we talk about new and emerging viruses. Today we're going to focus in on Rhabdoviridae, and what we're going to see is that these viruses within this family utilize a similar mechanism of replication of their genome. They have a similar genome organization, and once we learn for one family member in this order, the features are going to pretty much apply to the remaining family members. In the case of Rhabdoviridae, they have a really cool looking structure. As you can see from this EM on the left, they have really kind of a bullet shaped is really the best way to describe these viruses. That is, they have a flat end on one side and then kind of a curved, almost U-shaped structure. And this is due to the envelope that is surrounding a helical nucleocapsid, as we will see. Anyway, because of the structure of the virus, this is where the family gets its name, rhabdo meaning rod. They're kind of rod-shaped. When we look at this particular family, I mentioned at the outset that the virus that's most notable in this family is rabies virus. That's not really a virus you want to play around with so much in the lab if you don't have to. For the studies on the replication of the Rhabdoviridae, we use another less deadly member of the family, and that's vesicular stomatitis virus that's shown in this EM on the right. And you may see that vesicular stomatitis virus, sometimes it's abbreviated VSV, and that vesicular stomatitis virus would be the prototypical member in this family, where we've used that for laboratory studies to determine the replication scheme of the virus. Looking at the characteristics, the family characteristics of this virus is in class 5 of the Baltimore classification scheme, and that means it is a negative sense RNA virus. It's single-stranded, it's linear or non-segmented or monopartite. The genome is on the order of about 12,000 nucleotides in length, and that kind of varies just a little bit depending on the particular virus within the family. And this 12,000 nucleotides is about 50% larger than the last virus that we looked at, that was Picornaviridae, and therefore we expect that there might be more stuff going on with this genome. When we look at the symmetry, this virus displays helical symmetry. It is actually an enveloped protein, an enveloped virus, and within that envelope there is one virally encoded spike protein. It's much larger than the Picornaviridae, ranging from 75 to 180 nanometers in diameter. That's just going to be the length versus the width. In this particular family, huge family. There are more than 200 members in this Rhabdoviridae family, and they infect a wide range of organisms, from plants to insects, fish, and mammals. We'll be focusing in on the ones that infect mammals for our purposes, but it's just a really super large family of viruses. Looking at a little more detail of the structure of the rabies virus, what we see in this bullet-shaped appearance is that, as in this cutaway, we can see that there is a lipid bilayer here, and that lipid bilayer contains the envelope spike proteins, and then and that's proceeding on the outside of the virion. But if we look on the inside, just underneath the plasma membrane, what we find is that these viruses within this order utilize a matrix protein. We talked previously about a matrix protein, and here's our first example in a virus. The matrix protein then is going to be found in the virus, and then there is a little complex that is seen inside of the virion that contains the RNA, and this RNA is going to be RNA is in a helical structure. The RNA is going to be bound by proteins, one of which is the nucleocapsid. And what we see in these viruses is that they also have an additional protein. In this case of rabies virus, the additional proteins are called P and L. And if we look up at the top at the figure, we can see that the virus has this ribonucleoprotein. This is the capsid protein that's going to associate with the genome. And we're going to see that this capsid protein, which is called N for nucleocapsid, is going to be tightly associated with the genome. In addition to that, because this virus is a minus sense RNA virus, that means that it has to bring in its own RNA-directed RNA polymerase. And that's going to be the function or the reason that we have these additional proteins, P and L, attached to the complex. The L protein is the polymerase protein, and then the P is called a phosphoprotein, and that's going to be important and required for the replication as well. The genome then, the genomic RNA, has both the nucleocapsid or the N protein as well as the P and the L protein. And then I'll just draw our attention to the top, the envelope glycoprotein that is protruding from the surface of the membrane of the envelope is called G for glycoprotein. And we use these single letter designations to describe most of these proteins. We have the G protein, the P protein, the N, L, and the M. And that's going to be pretty much it for this virus. When we look at how this virus is going to get into the cells, what we find is that the glycoprotein, the spike protein, that is glycoprotein G, it has some sequence homology to some proteins that are made by snakes that function as neurotoxins. That is, they're toxic to the nervous system. And that discovery kind of led to the identification of the receptor for the rabies virus. Attachment of rabies virus initially is going to be through binding to receptors that are found in your muscles, and that's the nicotinic acetylcholine receptor. This receptor is, so sometimes it's abbreviated So nicotinic acetylcholine receptor, and this receptor is found on your muscle cells, and it's required for efficient contraction of your muscles in response to nerve signals. And that's going to be important for how the virus is going to be able to establish initial infection. And what we're going to see is following binding to the receptors, the virus is going to enter using a pH-dependent fusion with the endosomal membrane. It's going to be taken up by receptor-mediated endocytosis into the endosomes. In this acidic interior, the endosome, the nucleocapsid can now get out into the cytoplasm, and the M protein is going to move away from this little complex of the RNA and the N and the P and the L during this uncoating process. This kind of summarizes some of the initial steps, and we can look in a figure on the next slide. This slide summarizes the life cycle of the rabies virus, and what we're showing here is a neuronal cell, and this neuronal cell, as we looked at previously in our diagrams of spread of viruses through the nervous system, through the body, that is, we can cause a systemic infection either by moving through the blood, which is super fast, or we can spread through the nerves, or we can use a combination of both. We're going to see is the rabies virus is going to cause a systemic infection through the nerves. It doesn't get into the blood. It's pretty much restricted to the nerves. Okay, and that's why we're looking at this structure of a nerve. Here's a neuron, the cell body, and then we have the axon, and then we have down here the nerve terminal down at the very bottom. And the neurotransmitter would be released into the synaptic cleft, and then it's going to bind to the receptors that are found on the muscle cell. So here's a muscle cell, and here is the nicotinic acetylcholine receptor to which the virus is able to bind and get into the muscle cells. Well, that's fine for the muscle cells, and what we're going to see is that when you first come in contact with the virus is going to really get into these muscle cells. We will see that usually you acquire the virus through a bite, and that deposits the virus near the muscle cells where the virus can bind to those receptors, the nicotinic acetylcholine receptor, and get in and replicate. And we think, and oftentimes what we're going to see is an increase in the level of the virus at the muscle cell for a prolonged period of time. But then the virus is going to get out, and then it has to get into the neurons. The problem is that neurons don't have the acetylcholine receptor. They are the ones that are releasing the acetylcholine, and the muscle cell has the receptors. So the question becomes, well, how does the virus get into the nerves? Because it's going to get into the nerves, and it can replicate there, and it can make its way from the peripheral nervous system into the brain and the spinal cord and cause really devastating effects. In studies, recent studies, it has been shown that this virus, the rabies virus, is able to utilize a second receptor that is found on neurons. And this receptor is a receptor for a growth factor called nerve growth factor. Or NGF. And the virus is going to bind to this NGF receptor. And the NGF receptor is found at the synaptic terminal. And that allows the virus to be taken up into the neurons. And once it's in the neurons, now it's in the endosomes. And in the endosomes, we're going to, as a result of the decrease in the fusion, the virus enters by fusion, and in the endosomes at the low pH, we get the uncoating event. We can clearly see the uncoating following this attachment and uptake of the virus, and then now it's going to uncoat and be released in the neurons. This initial attachment, I should mention, is mediated either by binding to the nicotinic acetylcholine receptor or the nerve growth factor. The binding requires the glycoprotein G on the viral envelope. Once the virus is now in and uncoated, we're at the synaptic, at the nerve terminal here. And the problem is that the virus needs to undergo translation in order to produce the proteins. It's a -sense RNA virus. It has to undergo translation as the first step upon uncoating. It can do that because it has the polymerase, but the problem is all the other goodies that you need for translation are not found at the nerve terminal. That is, there's no protein synthetic machinery at the nerve terminal or anywhere along the axons. What that means is that the virus has to travel along the axons, and it does that using microtubule-based axonal transport that allows the genome to get into the cell body. And in the cell body, we have all the stuff we need. We've got your nucleus, we've got your ER, the Golgi, all of the components, the ribosomes, amino acids, everything you need to make proteins and replication are found in the cell body. The first thing then that's going to happen once the genome makes its way to the nucleus is that we're going to begin to see production of some messenger RNAs for translation of the viral proteins. So the first thing that's going to happen is that we have to go from our -sense RNA, so 3' to 5', and we have our polymerase with us, and through the actions of the polymerase, we're now going to begin to make positive sense messenger RNA. The first step that's going to happen then is transcription of the genome. That requires the L protein, which is the RNA-dependent RNA polymerase, and that's going to begin to transcribe the viral genome. And what we're going to see is this genome is linear, one long piece. We are going to make individual proteins, separate proteins, and the way that we're going to make separate proteins is because this virus will produce separate messenger RNA transcripts for each of the genes, and that each one of those messenger RNAs looks like a host messenger RNA, has a 5' cap, poly A tail, and can then be utilized for translation in the cytoplasm of infected cells. So each viral transcript is capped, polyadenylated by the viral L protein. And this is summarized in the next slide. If we look at the organization of the genome, shown here, and with these -sense RNA viruses, we're looking at going from 3' to 5'. The genome, about 12,000 nucleotides in length. We actually know exactly how many nucleotides. And what we're going to see is that in the process of transcription, the virus is going to produce 5 separate messenger RNAs. And the way that it will produce these 5 separate messenger RNAs from this linear genome is by a mechanism that we call a stop-start mechanism of transcription. The polymerase is going to start transcription, and then it's going to stop, and then it starts, and then it stops. And that will allow it to produce separate messenger RNAs. The way that it begins, it's going to start at the 3' end. And the polymerase, so if we just draw the L protein, it's going to sit down at the 3' end, and it will begin transcription. It's going to transcribe along until it hits a region that is identified here as a stop. This region is the stop portion of this genome. And what happens is that the polymerase goes along here until it hits this stop, and then often, and in the process, what it's producing or transcribing is a positive sense messenger RNA. And this positive sense messenger RNA has a 5' cap. The polyadenylation signal is red. It's going to have a polyadenylation or poly-A tail. This is a separate messenger RNA that gets released when the polymerase encounters this stop region. Excuse me. And this stop region is really a lot of secondary structure that causes this polymerase, this L protein, to be kind of destabilized a little bit. And oftentimes what will happen is that when the polymerase encounters this stop, it falls off. And what has to happen then, in order for us to transcribe the next protein, is that the L protein is actually going to go back. So it's fallen off. Here's the L protein. And it's going to make its way back over to the 3' end, sit down, and start over again. The first gene that it will transcribe will be the same gene that it already transcribed. And now we're making 2 copies of this messenger RNA. That's okay. This messenger RNA, the first messenger RNA, encodes the N protein, and the N protein is the nucleocapsid protein. We need a lot of this N protein, and by making another transcript, that provides another template for additional production of this N protein. Okay, well now we're on round 2, where the L protein is moving along and it transcribes the messenger RNA for the N protein, and again it hits this stop region. About 2 out of 3 times that the polymerase encounters this stop region, it falls off, but sometimes it's able to continue on. It's kind of shaky, but it's able to start again. And it's going to begin to move down the genome, and when it encounters the transcription start site, it will now begin to transcribe this second messenger RNA, and the second messenger RNA will encode the P protein. When the polymerase gets to the end of the gene, it reaches and recognizes the polyadenylation signal, adds the poly-A tail, and encounters a second stop. And this time, the same thing happens again. About 2 out of the 3 times, that polymerase, when it gets down to here at the second stop, it's going to fall off again. And if it falls off, instead of rebinding internally, which in theory it could, but experimental data has shown that for the most part what happens is that polymerase that falls off is going to go back to the very 3' end, start all over again. And that's okay. We're just going to scan along, transcribe, and now we're making a third messenger RNA for the N protein. And this process continues over and over and over again. Every time the polymerase gets to this stop region, the poly-A tail is added to the transcript that is being produced, but it oftentimes will fall off. And if it falls off, it has to go back to the beginning and start all over again. What that means is, for all of the members of the Mononegavirales order, their genomic organization is the same. That is, the N protein is the first gene closest to the 3' end, followed by, well, and I should say that the last gene in the gene order is the L, or the polymerase. And then there's a little bit of variation in the middle, but generally you will find next to the N protein, you will find the P protein, followed by the matrix protein, followed by the glycoprotein, and then finally the polymerase. What this means is that members of this order of viruses produces different levels of each messenger RNA within infected cells. We say that there is a polarity of transcription. We make lots of the N messenger RNA and very little of the L messenger RNA. But that's okay, because it turns out that this N protein, we need a lot of it, and it's very important. Of course, we need the L protein, the polymerase protein, but we've already brought a copy of it in with us. It's already functioning. We don't need a whole lot of this. We definitely need to make the polymerase protein to be packaged with the virion together with the RNA, but the absolute levels of it are, the requirement of it is much lower than it is for the N protein. So these viruses utilize this stop-start mechanism of transcription to produce a polarity of transcription products, and that generates a lot of the N protein and very little of the polymerase protein. Okay, here are, I put this in as a table just for you to be able to see what these proteins do. Here's the N, don't worry about the length. Here's the N protein, and as I said, that's the coat protein of the nucleocapsid. The P protein, which we haven't really talked about so much here. We'll talk about it when we talk about some of the other members in the family, but it's associated with the polymerase. The matrix protein, that's also part of being in the virion. The glycoprotein, the G glycoprotein, this is the one that binds to the receptor. And then finally the L, which is the polymerase. Sometimes you'll see this terminology of replicase. So it's the polyprotein with a DNA replicase. And it has the ability to add a 5' cap, methylation, as well as a poly A tail, depending on the needs of the gene. When we look now back at this diagram of the replication or the life cycle of the virus in the neuron, we were at step 5 previously, and in step 5 we were able to transcribe the messenger RNAs for each of these 5 genes. And these messenger RNAs, as I said, have a 5' cap and a poly A tail. Therefore, they look like the cellular messenger RNAs, and they have no problem being translated. They will be translated in different locations depending upon the protein. Most of them are going to be translated in the cytoplasm by ribosomes that are just the free ribosomal pool that's found in the cytoplasm. The N protein, the P protein, the M, and the L, they are all going to be translated in the cytoplasm. The G protein, though, that's a glycoprotein. Therefore, the glycoprotein will begin its translation in the cytoplasm, but very quickly it will be directed to the endoplasmic reticulum, where it will be translated by the rough ER. And that's because it's a glycoprotein. The G protein is going to be post-translationally modified, that is, it's glycosylated, and that glycosylation event requires translation in the ER, starting in the ER, and then going into the Golgi, where the sugar residues, that is the carbohydrates or the glycosylation, it will be modified. And that's shown by these little arrows, showing that the glycoprotein is going to make its way through the ER, through the Golgi, bud off into vesicles from the Golgi and make their way to the plasma membrane, where they will be inserted in the plasma membrane, waiting for the nucleocapsid complex to join up. In step 7, then, we're processing the glycoprotein in the ER and the Golgi using the standard cellular means for glycosylation. Okay, we've undergone transcription, and those transcripts have been translated either in the cytoplasm or via the ER and the Golgi. We've made then the proteins. Now what we need to do is make some of the genome, and we're going to enter into the replication. This replication is going to come from the same genome that we started with, that -sense RNA, and that creates a little bit of a problem for these viruses. When we have in looking at this diagram, when we have a -sense RNA genome, starting 3' to 5', and I've described to you that during the process of transcription, we produce these messenger RNAs by this stop-start method, where the polymerase will sit down, it will transcribe, and then it falls off. And then it sits down again, and it may transcribe a little further, but it falls off. And that's fine. That's a great mechanism for producing separate messenger RNAs that can all be modified to be appearing like the host messenger RNAs. But we have to use this is the genome, and we need to use this genome for replication using that same L protein. In order for this virus to replicate, we have to make a full-length positive sense copy. Right? For replication, we have to copy the entire genome in one piece. We can't have all these little fragments serving as the genome, because the genome is a linear genome. And that's, how do we do that, right? Because this polymerase, we've seen, it sits down and it falls off. It sits down, falls off. Sits down, falls off. What we're going to see with these viruses that are found in this order is that we have to somehow stop transcription and instead turn our attention to the process of replication, where we can produce a full-length positive sense or antigenome that can then be used as a template to produce the full-length -sense that can be copied into virions. We have to shift the focus of the virus from transcription to replication. And the way that this virus is going to do this has to do with the N protein, that very first protein in the genome that we make a lot of. Members of the mononegaphiralis are able to switch from transcription to replication because of the actions of the N protein. The levels of the N protein are going to control the switch from transcription to replication. And that will allow us to produce a full-length positive clone or strand that is covered or encapsulated by the N protein. If we look at the diagram down below, here is the genome that came into the host cell. The genome going from 3' to 5', that's the -sense copy, and it has a lot of that, some of that N protein bound to it. It has a certain amount of the N protein, enough that it will allow this process of transcription to occur, producing these 5 separate messenger RNAs. As we've discussed, though, we have this polarity in the production of these messenger RNA, and we make lots of the N messenger RNA, which means we make lots of the N protein. And as transcription proceeds, the levels of the N protein increase. And as the levels of the N protein increase, what happens is that those N proteins are able to go and bind and completely cover the genome. And when they can completely cover the genome, then as the polymerase sits down on the genome, it begins to copy, and now we're replicating. Normally, there would be these stop, and I'm just indicating those by S. But now with the increased levels of the N protein, the polymerase doesn't recognize that stop signal as a place for it to fall off. That is, the secondary structure, whatever was there that caused that polymerase to destabilize and dissociate from the genome, doesn't exist. And that allows the L protein to migrate and copy all the way down, producing a full-length copy that also is going to be bound by a lot of this N protein. And then that full-length genome, antigenome, can now be used as a template to produce full-length positive sense that will also get bound by some of this levels of this N protein. It's then this increase in the N protein that is going to mediate the switch from transcription to genome replication. And this really describes what I've just kind of showed you. After the N protein is made, bounds to the beginning of the viral genome, that called the leader, it's just at the very beginning. That allows the full-length positive sense to be made, and these are made, and as they're made, the N protein is binding and encapsulating, and that's called the antigenome. That antigenome is just the positive sense RNA. And this antigenome then serves as a template for making more progeny, more minus sense RNA, and those minus sense RNAs can now be packaged into the virion. For each antigenome that we have, we make about 50 copies of the minus sense RNA. So we make a few of these antigenomes in order to be able to produce enough copies of the genomic single-stranded minus sense RNA to be packaged together with the ribonucleic complex. Okay, we're back here at our diagram of the life cycle. We've processed the G proteins and they're in the plasma membrane waiting for the nucleocapsid. We've undergone transcription and translation of the additional proteins, and now we have replicated the genome. All we have to do is kind of build up enough level of these components. As soon as we have enough minus sense RNA, the N, the M, the P, and the L proteins, now we're going to begin to get the production of some mature virions, and these mature virions will then be able to bud from the host cell. And that assembly process, here got ahead of myself, in step 9 is the assembly process, and that assembly process requires the presence of the matrix protein. The matrix protein is shown in green, and that matrix protein, as you may be able to see, is just sitting there right next to the envelope glycoproteins, and those envelope glycoproteins, that glycoprotein G is shown in blue, and those are the sites of the membrane where the envelope proteins are found. And then as soon as we assemble all the other components, then that is the N and the P together with the L, we assemble all that together, and they will then recognize the matrix proteins, which have recognized the glycoproteins, and that begins this process of distension of the membrane that we will describe as budding, and then we get release of the virions. And these virions then are released, and because they're predominantly released by the cell body, these virions have the ability to interact with other neurons that are nearby, spreading the infection, and that can allow many of these neurons will make connections to the central nervous system, and that's going to allow this virus to go from the peripheral nervous system into the central nervous system, which is where it can really cause a lot of problems, as we will see in the next topic.